Hey guys, diving into Unit 4, chemistry and energetics of life. So we'll do photosynthesis, respiration, all the macromolecules stuff, how we build and break down molecules, things like that. So we're going to start with this question. And you've seen this question before. I've asked it of you a few times. What are the two things we need? Hopefully you can pause this and check with your neighbor if you like. I do recommend you watch these videos with somebody else, so that you can bounce ideas off of each other. And when these questions come up, you can talk to each other about them. So of course it's matter and energy that are required, and we get both these things from food. Oh, look at that. There we go. Food provides the energy for all the chemical reactions we need to make happen. It provides the energy for all of our movement and everything else that we do. It also provides the building blocks so we can make more of us, whether that means growth or repair or reproduction. We need those building blocks. We need amino acids to make proteins. We need nucleotides to make nucleic acids, things like that. Life is organized, right? It's because of this constant input of energy, we are able to maintain order. Whether we're a plant or an animal, energy is always coming in. That allows us to defy the natural tendency toward entropy so that we can maintain these ordered lives where we run around and eat baby giraffes and stuff. The inevitable result of the loss of order is, of course, a 17th century painting with heavy-handed symbolism. It's death, right? You lose the order, you lose the life. Chemical reactions of life happen in a couple of main categories. We can divide it into a couple of main categories. So 3 of these terms go together, and the other 3 of these terms go together. There are 2 separate groups here. Take a minute, pause the video, see if you can sort it out. Put these into 2 categories. Should be 3 terms in each category. So hopefully you got this endergonic, right? That's a reaction that requires that we put energy into it. We'll call that type A. And when we're putting energy into things, we're building things. When we break things down, that is a catabolic reaction, and that is going to release energy. That's an exergonic reaction. The penguin says anabolic steroids, right? You take anabolic steroids if you want to get swole. You shouldn't. Please don't. But you take those to get swole because you want to build muscle, right? Anabolic just means building. So breaking, catabolic, exergonic. When we break things down, we get energy out. When we build things, endergonic reaction, we put energy in to build things. We have 2 main processes that we use for building and breaking down. We use dehydration synthesis for building, and we use hydrolysis for digestion for breaking things down. So you should be able to sort this out. One of these is from group A that we were just talking about, and the other one is from group B. Check with your neighbor. Should be pretty straightforward. Synthesis means to put things together. Plus we got a picture here of 2 separate things being put together into one. We got another picture here of a single thing being broken into 2. So hopefully you're able to sort that out. So you can see that this is B and this is A. So I'm sure you know what macromolecules are, but just to double check, you might pause the video and make sure you know what macromolecules are and be able to identify the 4 major classes of macromolecules. So hopefully you know that macromolecules are just big molecules, big carbon chains with things that are attached to them to make it interesting. 4 major categories, of course, are lipids, carbohydrates, proteins, and nucleic acids. And we'll go through each one of these in the next lecture. So polymers are just many units, and so they're made up of monomers, single units, like a choo-choo train. A protein is actually chains of amino acids. A single chain of amino acids, we usually call that a polypeptide, and we'll hit that later, but it's like a bunch of cars on a choo-choo train strung together, one right after another, attached to each other in a chain. That's a polymer. Now you can stick molecules together in other ways that are not a polymer, but if it's repeating units of similar, monomers, then those are going to be a polymer. So you'll see that some of these macromolecules form polymers and some don't. When we build a polymer, we're going to do dehydration synthesis, which is that thing we were just talking about. So when you do dehydration synthesis, we've got a monomer here, and we've got this growing chain that we want to extend. We want to add this monomer to the chain. And so there's going to be a hydroxyl group, an OH, on one and a hydrogen available on the other. And we take those two hydrogens, that oxygen, we take all that out. Now, if you think about it, when I break this bond right here, I've got an electron that wants to form a bond. When I break this bond right here and I form the water, now I've got two places that want to form a bond because they used to have something stuck on there. I've got a single electron. And so what those do is go together and form that new bond together. So now we've got this other monomer added to the growing polymer. And in that process, we produce a water molecule. And that's why it's called dehydration synthesis. Water has to come out, dehydration. And synthesis just means to put things together, right? Syn just means together. This isn't going to happen by itself. This is going against entropy, that natural tendency to, spread disorder. And so this is going to require energy, and it's going to require an enzyme. Anytime you're doing a chemical reaction deliberately in your body, it's an enzyme that makes that reaction happen. Sometimes this is called a condensation reaction for the same reason it's called dehydration. It's just because water comes out. So you're going to need an enzyme and you're going to need energy to make this happen. When we're breaking down a polymer, the reverse of dehydration synthesis is hydrolysis. And hydrolysis is what we do in digestion. This is what happens in your small intestine to all the proteins that you eat. They get chopped into individual amino acids. All the starches you eat get chopped into individual glucose molecules. So we do this hydrolysis. And the word root lys, L-Y-S there, that means something's breaking or cutting. And hydrolysis, right? Hydrolysis, we're using water to break something. So we're just doing the reverse of what we did before. We had this bond that we made in dehydration synthesis. We're now going to break that bond, and we're going to have those two dangling electrons again that want to form a bond with somebody. We split this water, we lyse water, and we split this monomer off of here. We stick a hydrogen on one, we stick a hydroxyl on the other, and now we've got happy bonded electrons, nobody left dangling, and we've split water. So we've used one water, and we've taken off our single monomer. We have different enzymes that do hydrolysis depending on what the compound is. We have some that will just chop it somewhere in the middle, and others that will only take off the terminal one of whatever the polymer is. But either way, they do this hydrolysis. They use water to split. And it's just the reverse of dehydration synthesis. But you're still going to need an enzyme, right? Every chemical reaction that's going to happen in your body in any kind of deliberate and meaningful way is going to have an enzyme that catalyzes that reaction and makes it happen quickly. So don't pay much attention to the little green circles because those aren't really representative. If anything, they're a little incorrect for biological purposes. At least one of them is. But looking at these graphs, we're looking at delta G, and that's really telling us whether something is endergonic or exergonic. That's what we're seeing in this graph. So you should be able to tell by whether there's more energy in the product or in the reactant, which of these reactions is happening. So let's do a couple of different things. One, for these 6 terms again, which graph represents each term? And 2, which one of these graphs represents dehydration synthesis in biology or hydrolysis? One of them is hydrolysis, one of them is dehydration synthesis. And you should be able to get there from that. So pause the video, make sure you can address all that. Which terms go with which graph, and which one is dehydration synthesis, and which one is hydrolysis? So hopefully you see that we're ending up releasing energy. We have less energy in the products in this graph down here. And that means we are breaking things down. If we're breaking things down, it's breaking, it's catabolic, it's exergonic, energy coming out. So this one is hydrolysis. And here we've had to put energy in. So we're building, right? So we're endergonic, we're building, we're anabolic. And this is dehydration synthesis. So hopefully that's pretty straightforward. Of course, the way the graph goes up and then comes down, we need to talk about that. That's activation energy. That'll come up later when we start talking about enzymes. It's also in this unit though. So we can take H2 gas and O2 gas, react it together with a spark and produce water and release a huge amount of energy. We do a similar type of thing with hydrogens and electrons. Instead of hydrogen gas, we react that with water eventually. But we do it in a much more stepwise fashion so that we don't explode. I think you can all agree that not exploding is always a good thing for living things. And we do this in a very controlled, stepwise fashion, and it allows us to generate ATP. So it's lots of complicated steps so that we don't get all the energy at once, because that would be too much energy for our cells to withstand. So we do cellular respiration to get energy out of these things, and we go stepwise and very slowly and release that energy in a way that is not damaging to our cells. For example, this is glycolysis. This is the very first thing we're going to do with the glucose molecule. We're starting here with the glucose molecule, And you can see we stick a phosphate on here. We do some rearranging. We eventually split this guy in half, and we end up with 2 3-carbon molecules. You can see 1, 2, 3 carbons, and 1, 2, 3 carbons. Glucose is 6, right? C6H12O6. So we've got 2 basically identical. They're isomers of each other, halves of this glucose molecule. So we took this 6-carbon molecule and split it into 2 3s. And then we just have an isomerase, which is what this says right here. This is an enzyme that turns this isomer into this isomer. And now this guy can continue twice along the rest of this pathway with once with each half. And you can see we have to use some ATP to make this happen. We have ATP going in, we have ATP going in, but we also have ATP coming out twice. And remember, we go through this path 2 times. So we get 4 out. We put in 2, we get out 4. But we do it stepwise. Again, we're not exploding our glucose. We're releasing our energy in very controlled ways so that we can use these ATP. So we're generating ATP doing glycolysis. And every living thing on Earth does this. It used to be an AP, you'd have to know all these enzymes, you'd have to know all the intermediates and things like that. We don't really need to worry about that now. We do need to know that we end up with pyruvate, but we'll talk about that later. We start with glucose, we end up with 2-pyruvate. That's glycolysis. We also generate some ATP, and we generate NADH, which we'll talk more about that guy later. Pyruvate comes to the mitochondria, and this is not something you need to worry about right now. We'll go through some of this later, but I'm just trying to show you that we break down these fuel molecules, these energy molecules. We break them down stepwise to release energy. It's very, very controlled, and we're generating NADH. This GTP is going to be used to make ATP. We're getting more NADH. These are all processes that make energy available to us, right? Here we're getting some ATP also. So we're getting a lot of energy out of breaking down this glucose, and we're not getting it like an explosion. We're doing it all stepwise. So we eat high energy organic molecules, proteins, lipids, carbohydrates especially, right? Those we break down in our digestive system into monomers, into building blocks or pieces of some kind. And then we absorb those. And then we can use those building blocks, either break them down further for energy, or we can use them to assemble parts of ourselves. Inside of cells, of course, we have to use energy all the time. And we have this really usable molecule. You probably already know what it is. It's like money. It's like cash. It's usable right now. It's ATP. I will refer to ATP as the boom because every time it's going to make that sound. Indeed. All right, so adenosine triphosphate, this is ATP. It's basically, this is a nucleotide. If you look at this guy right here, that's a nucleotide of RNA because this is ribose, this is adenine, right? Remember adenine, uracil, guanine, cytosine. Those are the nitrogenous bases you find in RNA. But adenine with the ribose and then 1 phosphate stuck on here, that would be 1 nucleotide of RNA. But with 3 phosphates stuck on there, that's ATP, triphosphate, right? Adenosine, here's your adenosine, triphosphate. So we have adenosine monophosphate, that's 1 phosphate, diphosphate, and then triphosphate. But generally what we do in our cells is we take the ATP, we pull a phosphate off when we need the energy, and we use that phosphate, or we use the energy in that bond. And so we make ADP and what we call inorganic phosphate, which is a lot of times just abbreviated P sub I. So we take adenosine diphosphate and this inorganic phosphate, and in the mitochondria, we make it back into ATP. Then we go use the ATP, make it into ADP, and release that inorganic phosphate again. So we get this cycle going where we're constantly replenishing our ATP. And that's our usable form of energy. When we're doing things inside of cells, proteins need to be changing shapes, need to be doing things. We do that using ATP. That's our cash. That's our use right here, right now energy. It's extremely reactive stuff. We can't move this around in the blood because it would react with everything in between the 2 places you were trying to send it. So it's not a transportable energy, it's a usable energy. Why does this have so much energy for such a little molecule? Well, it's those 3 phosphates, right? Phosphates are a negatively charged polyatomic ion. So I've got this negatively charged polyatomic ion, another one, and I shove it right next to it. They don't want to be next to each other. They're both negatively charged. They're trying to repel each other. But if I can get them close enough, I can get a covalent bond on there. They're stuck, but not happy. And if I grab a third phosphate and stick that one on there, I really have to kind of cram it on there because it doesn't want to go. I got 2 negatively charged phosphates, and a third negatively charged phosphate is like, no, I don't want to be there. But I can click it on there, and now I've got ATP. And you can think of it as being spring-loaded, right? That third phosphate is just ready to pop off. So an enzyme is going to grab a hold of this thing and be able to take it and use that energy to make something happen because it's got energy just hanging by a thread ready to just explode off of there. So it's an unstable molecule, but that's what makes it so useful. Diphosphate. Muppet newsflash. Triphosphate. Adenosine triphosphate. Probably should have made that AMP go away. Okay. I like this little diagram. It's super simple, but it does kind of tie it all together. So here's what you eat, and here's what you make. This is just tracking the chemical energy. We also have, of course, building blocks that we'll need to talk about at some point. These small molecules, those also come from digestion, from our food that we eat. But we're going to take glucose. We're going to do this exothermic reaction, which releases energy. We couple that with this endothermic reaction. So the energy from this is used to make ADP and inorganic phosphate into ATP. So coupling exothermic with endothermic reactions, this is a very typical thing that we do in biology. This is what we have to do. We want to do something. We need to break something down to get the energy out so we can make that thing happen. And again, here it is again, where we're building large macromolecules, making ourselves, making our enzymes, our proteins. We do that from the energy from this ATP. And we got that energy from the food that we ate, and we coupled it with a reaction to generate that ATP. So this diagram really sums it up nicely. So make sure you have a look at everything on here, and it should all make sense. So just to kind of get a big picture on our energy budget, right? In living things, our energy is chemical energy. We're talking molecules with energy trapped in bonds. A lot of them are carbon-hydrogen bonds, but what we really end up doing is oxidizing carbon. We'll talk about the redox reaction that occurs there in a bit. But this chemical energy, right, is originally trapped by some plant somewhere. Some plant did photosynthesis and stuck energy, trapped energy that came as light, turned it into chemical energy. Remember, energy is not made or destroyed, but we can convert it into different forms. And that chemical energy can then be passed on from organism to organism through the food chain. What we use as immediately usable energy, we've talked about this is ATP. This is a main one. We do use a couple of others. There's GTP and TTP and things like that, but ATP is definitely the main one. I mentioned that ATP is not transportable. We can't move it around inside our bodies because it's so reactive. It'll react with something along the way. And so multicellular organisms need to be able to transport energy that isn't ATP. And we usually use glucose. As animals, we use glucose. That's our primary source of energy that we move around, our transportable energy. In plants, so this is in animals, in plants, it's typically sucrose. So in this little lady, she uses glucose to transport around. She's kind of grumpy. But plants are going to use sucrose, which is a disaccharide. It's a little bit bigger molecule. We'll talk more about that later. When it comes to energy storage, plants and animals also do that differently. We do a couple of different things as animals. Typically, we make a molecule called, and we'll talk about all of this later. I'm just trying to get some big picture on there now. But we use a molecule called glycogen, or sometimes it's just called animal starch. It's a lot like plant starch, but it's more complicated. It's more branchy. And we'll talk about why. And then long-term storage, we will make fats. We'll make triglycerides and store those. That's for our very long-term storage. In plants, you're going to use just what's called starch, which is just chains of glucose. Glycogen is chains of glucose too, but they're different. Everything on this page we'll talk about again later. I just wanted to kind of get a big picture on this. So now we're going to look at energy on an organism level. Different organisms kind of manage their energy in different ways. When we're looking at animals, we have endotherms and ectotherms, and we've mentioned this before, and you probably remember what these are. But endotherms are what we usually call warm-blooded, and ectotherms are what we call cold-blooded. Endotherms regulate their own body temperature like you do. You're always 98.6 or 37 degrees Celsius. Other animals have slightly different temperatures, but really, they're usually pretty close to that. That's a good enzyme activity temperature. Ectotherms rely on their environment to provide their temperature. So they, They sometimes will have behavioral things that make it so they do things to warm up. They will go lay on a warm stone or something like that, or bask in the sun so that they can warm up and get their enzymes functioning properly and everything, get that chemistry happening that's necessary for life. But each one of these has its consequences, its pluses and its minuses. If you think about endothermy, you should be able to stop the video and look at both ectothermy and endothermy and think about what are the advantages and costs of both. So if you want to pause the video, talk to a neighbor, somebody, talk to your dog, whatever, sort out what's the benefit of being an endotherm, what's the disadvantage, what's the benefit of being an ectotherm, what's the disadvantage. So hopefully you figured out that the advantage of being an endotherm is that you can be active all the time. You don't have to wait to be warmed up by the sun to be able to do all the things you want to do in a day. So they are always active and never vulnerable because of that. The cost is, of course, that it's a huge calorie deficit. I mean, we spend a huge amount of calories just keeping warm, running our normal metabolic processes, and at the same time, maintaining our body heat so that all those enzymes are functioning when we want them to be. Most of the calories you burn in the day are just doing that, right? Even if you went and ran a half marathon today, the bulk of the calories you burn today would not be in that half marathon. It would be all the rest of the day just doing your normal day-to-day stuff. If you're an ectotherm, the advantage, of course, is that it's super cheap. It's very low in calories, and we'll look at that a little bit later on. But the cost, of course, is you're vulnerable. I mean, this lizard here was easy to pick up because it was barely moving at all. When it's cold, ectotherms are very vulnerable because they can't do chemistry, so they can't do all the stuff that's necessary to, run around, protect themselves, things like that. So we've got a graph here that shows two different lines, and we're plotting ambient temperature, the environmental temperature, versus body temperature. You have to figure out which is which, and this should be pretty straightforward. So take a minute, pause the video, make sure you can tell which one's an endotherm, which one's an ectotherm. The blue line is one and the red line is the other. So hopefully you can see that the otter here is the red line and the fish, the bass, is the blue line. And at low temperatures, right, this guy has a low body temperature. And it doesn't matter what the temperature is, this guy's body temperature is pretty much consistent the whole time. So that's an endotherm. An additional question would be, who's spending more energy where? And so if you think about that, if we looked at a graph that had how much energy is being expended by each of these organisms at different ambient temperatures. So take a second to think about that. What would this graph look like if the body temperature was not your y-axis, but was amount of energy expended per hour, let's say, for each of these organisms? What would that look like across these different temperatures? Pause the video and see if you can answer that. So hopefully you figured out that if you're looking at this bass, this ectotherm, this cold-blooded fish, at low temperatures, it burns very little energy because it's inactive. It doesn't really do anything. Whereas this guy at low temperatures is actually burning more energy than it would be normally. So it would have a graph that slopes down because it's going to spend more energy at low temperatures trying to stay warm than at high temperatures. So hopefully that makes sense. If you look at over time spans, like the maximum metabolic rate, in any given second, right, you and me and an alligator can burn a huge amount of energy. But over time, as we experience more cold periods and things like that, over a week's time, we burn a lot more energy than an alligator does because we're spending a lot of our energy just staying alive, staying warm, whereas they are saving all that. But at any given second, an alligator can move very quickly and have a lot of energy available to it. So this should be a pretty straightforward question. I think you could answer this pretty easily because reproduction is tricky, right? It's a big investment. It's something you really have to juggle when you're going to do this. So why do birds always lay their eggs early in the season? That's a question you should be able to answer. Now, looking at these organisms, just looking at the overall energy budget, it's kind of interesting because you can see we're a lot bigger, so we have a lot higher amount of energy that we use than some of these smaller organisms. But this guy is 4 kilograms and this guy is 4 kilograms, and we can see the difference between endotherms and ectotherms, over a year's time when we look at these. I mean, there's a lot more energy spent here than with this snake. What's also interesting is you look at how this pie chart changes for a very small organism. I mean, it's different for us and them. We spend a lot more on our basal metabolic rate. they spend a lot more on temperature regulation, just staying warm. So this is the part that we spend just to stay warm. And you can see a penguin, it's a little bit more than that. They do live in a colder environment. But these little bitty guys, it's a lot more, and that's because their surface area to volume ratio is so different. They're radiating heat out. They have to constantly be cranking out the heat because their surface area is so great compared to their volume. Whereas us, our surface area is much smaller, so where we lose our heat is a much smaller area per volume. It's interesting. It's not that big a deal. It's not something you need to worry about, but it's interesting. Looking at this bottom graph here, you can see that the energy expended per mass is much, much greater for this little bitty guy because he's spending, again, so much energy just trying to stay warm. And then if you compare the two 4-kilogram animals, the ectotherm and the endotherm, you can see again that the energy expended per unit mass is much, much larger for the endotherm. And that's pretty straightforward. And again, the human is actually less per kilogram because they're so massive and they don't lose so much heat because of the small surface area to volume ratio. So this is Franklin and Zwiebel. Zwiebel is a hairless cat. Franklin is 17 pounds. Zwiebel is 7. Why? Based on energetics, but based on what we've been talking about, why is this cat so much smaller? They probably have fairly similar diet too. So pause the video, see if you can answer that. If you look closely, there's a lot of blankets on this bed. So this is a place that gets cold. And Zwiebel here has no fur coat. So he has to spend a lot more energy keeping warm because so much heat is radiated through this skin. Whereas Franklin here has got so much fur, he helps trap that heat with the insulation and therefore doesn't have to spend as much energy maintaining its body temperature. So he's able to bulk up eating the same amount. Zwiebel just spends a lot more of that energy staying warm. This is a fairly straightforward question too. So go ahead and read this, pause the video, and see if you can answer this question. So if you think about this, if you were to draw a graph on the amount of energy at any given time spent by these guys, and over time, here's their normal summertime, fall maybe, and then when winter comes, they go into hibernation. So they have this much lower energy expenditure because they're barely moving at all. Their body temperature drops. And then when spring comes, they warm back up and go back to this. But if they wake up, right, they lose this hibernation advantage and they get these spikes every place they wake up. And so they wake up. If they're periodically waking up, they're spending more energy. The amount of energy, the total energy spent is this area under the curve here. So they can end up dying from this because all the energy they've saved up, that's supposed to last them through the winter. But that only works if they maintain a very low level of expenditure in hibernation. So every time they wake up, their metabolic rate goes up, and that makes them burn more energy than what they normally would. And so therefore they don't make it through the winter. So that's the introduction to this energy thing, and introduction a little bit to the metabolism there too. So we'll go farther with this and we'll look more closely at some of these macromolecules and how they work in the next lecture. Hang in there, guys. You're doing great.